Good afternoon, doctors from across the globe. I'm Dr. Rajiv Kovil. I'm moderating the session along with Dr. Sheila Krishnaswamy, who has got the warm privilege to introduce Professor Anoop Misra. Over to you, Dr. Sheila.
Thank you, Dr. Kovil. It gives me immense pleasure to introduce a very well-known personality in the field of endocrinology, Dr. Anoop Misra. Dr. Anoop Misra is a member of the Academic Board of British Medical Journal. He is an associate editor of the Journal of Diabetes USA. He is an honorary professor, Institute of Life Sciences, Hyderabad. He is also a Padma Shri awardee by the Government of India. He has several other credentials. Due to lack of time, I won't be able to read everything. So over to you, Dr. Anoop Misra, to begin your presentation.
Thank you, Dr. Krishnaswamy, and thank you, Dr. Kovil. The topic for today is the recent concepts in adipocyte biology and focus on brown fat, and particularly implications for Indian population. Now, in this presentation, I will discuss white adipose tissue, beige adipose tissue, brown adipose tissues, structure, biology, and clinical implications. Now, this is a very, very exciting area of research currently, and I will make it very simple for our audience. Implication of these data will be discussed in context of Asian Indians. I have no conflict of interest, and this photograph shows my research team, an extended research team, including Dr. Naval Vikram, who will be speaking subsequently. The first part of my presentation, types of adipocytes and adipose tissue compartment, will just give you a background idea as to where we find which type of adipocyte, and then subsequently what is the function of it. Now, you see on the left side is the white adipocyte. And where are these found? This is the intrathoracic adipose tissue, and this is the intra-abdominal adipose tissue. Together they are called visceral adipose tissue. It's not only intra-abdominal adipose tissue is called visceral. And in this, in the abdominal adipose tissue is of particular importance. Now, it may consist of subcutaneous adipose tissue or intra-abdominal. Now, intra-abdominal is divided into various other areas. I'm not going to details of that, but both these compartments, subcutaneous and intra-abdominal, are important for metabolism. On the left side, you see brown adipocytes. I'll come to the structure in a moment. What is the difference between white adipocyte and a brown adipocyte? Now, these are found usually in hibernating animals, and also recently use of PET has shown that it is found in human beings. Now, these are not as found in as many areas as white adipose tissue, but you can see here, around scapula, supraclavicular area, perirenal area, and paraspinal area. These are the usual areas where brown adipose tissue. On this particular photograph shows side by side white adipose tissue and brown adipose tissue. These you can see very well. These have a little different structure, and I'll come to that in a little while. Now, just a little bit background information on the effect of gender and age on adipocyte, which is important to understand how the distribution of adipocytes and the type of adipocyte change over a period of time. In this respect, it is particularly important to consider the role of estrogens. Now, estrogens are actually anti-obesity agents. On one hand, they increase the energy expenditure by increasing sympathetic nervous system activity, and also by increasing natriuretic peptide, they convert the white blood cells to bright or beige fat cells. They also decrease food intake a bit. So after menopause, what happens? The 24 hours energy expenditure is decreased, so mainly sleeping energy expenditure, and the food intake increases after the menopause. So what happens as far as fat distribution is concerned? Now here, android and gynoid type of fat distribution, especially abdominal, are given. Uh, Women have more of a subcutaneous fat. You can see more of a subcutaneous fat. Intra-abdominal fat is also there, is not as much as in men. But as the menopause approaches, the estrogen decreases. This distribution goes to the android type of distribution where there is a lot of intra-abdominal fat while keeping the subcutaneous fat. At the same time, if you see the adipocyte, the structure changes from a smaller adipocyte to bigger adipocyte, and these are so-called sick adipocyte. I'll come to that again in after a couple of slides. As far as aging and adipocyte biophysiology is concerned, the predominant cell otherwise determining the fat accumulation in the body and its distribution is pre-adipocyte. As one ages, there is decreased ability to store lipids, decreased adipogenesis, and increase in the inflammation. This pre-adipocyte changes to so-called mad cells, which accumulate in ectopic tissue. At the same time, there is an increased amount of accumulation of free fatty acid, which adipocyte become larger. I showed you in the previous slide the image of large adipocytes. And these then are accumulating along with macrophages and then increase free fatty acid, and insulin resistance is the consequent phenomenon. So this is what happens in aging, and I already showed you about the gender dimorphism of the adipocytes and how the redistribution occurs once a woman goes into menopause. Now let's look at the functions of white and brown adipose tissues. This slide shows three arrows. One is white. White we know very well. All of us know. They are dynamic buffer for free fatty acid flux, and fasting state they release the fatty acid, and fed state absorb fatty acid. So free fatty acid comes predominantly from white adipose tissue. Have a lot of endocrine functions like hormone, adiponectin, resistin, leptin, etc. being secreted by adipocyte, and they have important function as far as the general physiology and metabolic physiology of body is concerned. And of course, they secrete a lot of adipokines. I'll come to this again in a minute. Now, as far as brown adipocytes are concerned, they increase thermogenesis and may increase satiety. I'll show you one slide on that. Beige are somewhere in between, intermediate. They are found a bit in white adipose tissue, and they also, once they are increased in number, they increase the thermogenesis. Now let's look at the dysfunction of the white adipose tissue. Now this slide may be a busy slide, but on the left side you see the white adipose tissue with the big droplets, and on this in the middle is the co-localization of adipocytes and macrophages. You see this, the smaller ones are macrophages, and the crosstalk lead to amplified paracrine environment. It's more clearly seen here, where these are the macrophages and these are the adipocyte, and they increase NF-kappa B activity, marked pro-inflammatory environment, and these are also atherogenic. So once there is a more accumulation of fat in the white adipose tissue, the free fatty acid, inflammation, they increase, and increased amount of free fatty acid and glucose goes all over the body and gets accumulated in various organs. Now this is helped by increased energy intake and lipid overflow. Now these organs can be abdominal, these organs can be liver, heart, muscle, kidney, and pancreas. But as far as our team is concerned, we have done large amount of research as far as abdominal adipose tissue is concerned, and particularly relevant to the subcutaneous adipose tissue. You can see here two compartments in the subcutaneous adipose tissue, deep compartment and the superficial. To us, this posterior deep compartment is what is metabolically important. We have number of studies. I'm not going to go into those studies. This deep compartment is also seen separately here. Using a software, we can segregate that. To our group and Naval's group, this is also very, very important. So together we have published a large number of papers as far as fatty liver are concerned. So excess fat deposition in the subcutaneous and intra-abdominal tissues and excess fat deposition in the liver, and with expansion of fat, which is very, very important because this expansion of fat increases H1F1 alpha. Now, this is the recent concept. This is an oxygen sensor which is upregulated. It is pro-fibrotic and inflammatory and decreases adiponectin. I think this has become of increasing importance and increasingly being researched. In general, adipocytes become dysfunctional. Now, what is a dysfunctional adipocyte? You know, look at the cartoon here and they say, this adipocyte say, help, I can't breathe in this particular obese person. This is a fat adipocyte. Increased subcutaneous adipocyte in obesity leads to hypoxia, which leads to fibrosis. Decreased sensitivity to insulin, insensitivity to insulin induced inhibition of lipolysis, more of free fatty acid, fat overflow, and lipotoxicity. So this so-called sick adipocyte is more often seen as far as Asian Indians are concerned. And this is my colleague from Dallas, where I worked, Dr. Manisha Chandalia's work, elegant work in PLOS ONE in 2007. Nobody has been able to better this work. And you can see the adipocyte in microns. Here on the left side, white Caucasians, right side, South Asians, and you can see large dysfunctional adipocyte, subcutaneous adipocyte from South Asia. And they are more likely to co-localize with macrophages and produce inflammation. Now, this is something which some of the investigators have hypothesized. On the top is the diagram of a person who's metabolically very good. Now, if there is excess energy, there is maybe some adipose tissue hyperplasia. Hyperplasia means increase in the number. And it may be in superficial subcutaneous tissue or deep subcutaneous tissue, but doesn't expand it much. And also there is not much of hepatic liver fat accumulation, and hence little change in the cardiometabolic status. The moment adipocyte size increases, not the number, size increases, and which happens in Asian Indians. Now, excess energy, there is increase in the adipocyte size in the superficial subcutaneous adipose tissue, deep subcutaneous adipose tissue, as well as intra-abdominal adipose tissue, which is seen also on this side in a research study which was done earlier by us. Also, they increase free fatty acid flux and increase hepatic fat accumulation, which may cause asphyxia, low adiponectin levels, and number of cascading metabolic events leading to diabetes and cardiovascular disease. So this is the hypothesis which has been shared in a publication by Dr. Sonia Anand and her group. Let's have a look at the brown adipose tissue. Now, brown adipose tissue and white adipose tissue have different origin. Now, look at on the top is dermomyotomal precursor cell. One side it leads to skeletal muscle development, other side it leads to brown adipose tissue development. Now, on the bottom is mesodermal stem cell. On one side it leads to beach, which is intermediate between brown and white adipose tissue, and other side to white adipose tissue. Now, there's a great amount of cellular plasticity here. White adipose tissue can go on to develop beach and beach to that. And this has tremendous application as far as research is concerned, as far as physiology of obesity and diabetes is concerned. Now look at the different structure of the white adipose tissue. Big one, like an egg with a yolk in between. And a brown adipose tissue, multiple small fat globules with a huge number of mitochondria which contain UCP1, which is very, very important as far as energy expenditure is concerned or thermogenesis is concerned. which is shown in this particular diagram. Now, the moment we are exposed to food of some sort, a cold, beta-3 adrenergic receptor is activated here, and which leads to increased amount of triacylglycerol being available to mitochondria. And on the right side, you see the UCP on the one side and long chain PUFAs on the other side, which lead to uncoupling and energy thermogenic. This apparatus is thermogenesis, and which may lead to energy expenditure. On the other side, beta-3 adrenergic receptor, they increase production of UCP1. So both sides, it leads to increase in the thermogenesis and then subsequently other events which may be beneficial to our body. Now, this brown or beige adipocyte has crosstalk to multiple organs, including CNS through catecholamines, immune cells, liver, through myostatin in skeletal muscles, and heart through BNP. So it is something which has connection to almost every organ in the body. So we summarize this information here. The brown adipose tissue usually found in hibernating animals. It has remarkable capacity to dissipate energy via brown fat-specific UCP1. Now, 18 FDG PET scanning shows that human beings, 10% of the population, are BAT positive, and maybe 90% are negative, and some intermediate. It higher at young ages, decreases with age. Amount of BAT is inversely proportional to BMI, adiposity, and intra-abdominal fat. Now, browning of white adipose tissue contains some beige cells, may increase thermogenesis and protect against obesity. This is of increasing research interest as far as obesity is concerned. Now look at this. This is the white adipose tissue, hypoxia, and fibrosis. This is the beige adipose tissue here, and it does not lead to fibrosis. So H1F1 alpha pathway is not activated here. So there is additional importance of beige or brown adipose tissue. Now, what does it mean for as far as Indian population is concerned? If you see here, clearly in this particular case control observational study, which was published in Lancet, South Asians have lesser total BAT volume as compared to white Caucasians, which means their resting energy expenditure, while you are resting and a white man is resting, our energy expenditure is lesser. We are going to gain weight. And non-shivering thermogenesis is much more increased with white Caucasian. So we are that way metabolically deficient as far as brown adipose tissue is concerned. Now, activation of brown adipose tissue is metabolically beneficial. That much we have come to know till now. That once we activate beige adipose tissue or beige to brown adipose tissue, it is metabolically beneficial. And this is shown in this slide. BAT significantly contributes to whole body thermogenesis, 200 to 400 kilocalories per day under cold conditions, which is a huge amount of calories, and also contribute to diet-induced thermogenesis. Indeed, BAT negative subjects in their 40s possess approximately 6 kg more weight. So BAT negative, more weight than BAT positive subjects. Hence, it is tempting to propose BAT as target for weight loss. Now, look at this particular human experiment where it is shown the once the BAT is activated. On the right side is activation of BAT here in black and white, and on PET here. So once patient is the person is just the temperature is neutral versus temperature is cold, there is improvement in peripheral glucose uptake and insulin sensitivity as shown here. The glucose uptake is much better once the person is cold and exposed to cold, and also there is activation of BAT. So this is clearly important as far as glucose also in lipid metabolism is concerned. So how can BAT or beige fat can be activated? Now this has practical implications, some of which is still confined to experimental studies. Here it says, well, I have to go to cold places to lose weight. This guy says, I have to visit every day. Well, this may well be true. Look at this experiment where the bat detected subjects were exposed to cold here. On the left side is the cold. And look at after some time, 6 weeks, the cold exposed subjects had markedly low body fat mass. Though their weight may not be less, but fat is decreased and controlled people did not. Now, the other way is to eat chilies, Capsaicin, you look at the capsaicin here. Same experiment, 10 people for 6 weeks exposed to some amount of capsaicin which they took and body fat decreased. So there are, apart from cold, there are other methods to increase fat in people. Another method is the coffee or catechin, which is found in coffee and the tea. Acute single dose of 615 milligram of catechin and 77 milligram of caffeine. Again, patient or people with high BAT versus people with a low BAT. Look at that, BAT, high BAT here. In acute study, the energy expenditure increased only in those people who had high brown adipose tissue, and rest it did not increase. And in chronic, if they were getting chronically for 5 weeks, again the same thing, cold induced thermogenesis increased in those people who had a high BAT. So you have to have a high BAT. High BAT can be induced also through certain substances. Those with high BAT have advantage and they respond better to certain substances. Now here this lady says take a lot of apple. Makes sense because peel of the apple contains a substance called ursocolic acid. Now this is the mice study which shows that if the mice are given ursocolic acid and compared to high fat diet mice, the brown fat increases tremendously and the relatively UCP levels increase and body temperature and energy expenditure increase. So there are other things also which are being explored which can increase this brown adipose tissue and make you lose weight. And some of these things are listed here. So physiological factors, cold, beta-3 adrenergic activation, endocannabinoid receptor activation and estrogens. Tea, catechin and coffee, capsaicin, ursocolic acid and resveratrol. Drugs, melatonin, and you can see melatonin on the left side, no melatonin, on the right side melatonin, and you can see activation of BAT cells here. Sildenafil, but unfortunately pioglitazone in clinical studies did not, sitagliptin did not, and fluvastatin did. And these are only some of, there are many more. I have just picked up a few, which means this is the active area of research and will be very important. So I'll just be finishing in 2 minutes. The appetite control, there is some element of appetite control with the with the activation of BAT, which causes increased meal-induced thermogenesis. Now, as far as adipose tissue metabolism, now I summarize here. White adipose tissue is increased free fatty acid, low adiponectin levels, insulin resistance, increased adipocytokines, and possible hypoxia and fibrosis. This is what is occurring as far as Indian population is concerned, with increase in the white adipose tissue. And there is decrease in the BAT, decreased energy expenditure, and possibly low satiety. And we have to actually activate beige and bright fat, and this should be attempted with exercise, diet, and various things that I have told you. So this is my final slide. There are gender, age, and ethnic differences in adipose tissue and adipocytes and related biology. The specific physiological function associated with white, beige, and brown adipose tissue, which have implication for obesity and diabetes. Role of various drugs, dietary factors to increase BAT and browning of BAT is increasingly being researched. Asian Indians have expanded adipose tissue stores, white adipose tissue, sick adipocytes, hypoxic adipocyte, predisposing them to early this metabolic state, and white adipose tissue should be lesser in Indians and bad should be more. More recruitment of beach adipose tissue is required, and these are exciting area of research. Thank you for listening, Vineet. Thank you, chairpersons, for chairing this session.
Thank you so much, Professor Mishra. That was a fantastic presentation. We have a few questions from people who are viewing this across the globe. I would start off with one of the first questions which came in. So this selective distribution of fat in the vascular compartment and the visceral compartment, is it driven by the brain and hypothalamus, or is genetic? Or why do some people like me have more abdominal fat and some people like you who are so thin and lean? So is this genetics? Is this the brain and hypothalamus? What exactly is the mechanism? Why does some people have brown adipose tissue and some don't?
Yeah, I think it's a combination of factor. Number one, as far as Indians are concerned, possibly centuries of problems that we had as far as nutrition is concerned, uh, famines, etc., food deprivation, which led to our muscles, they shrank, and the body tried to store a lot of fat, especially in the compartment it could store, like abdominal liver. So even one is so-called obese, non-obese person, even then a person has more of abdominal fat, more of subcutaneous fat, more of liver fat. But it is not so simple. It is driven by many other factors, and genetic is one of the important factor. Though I would say the importance of genetics is around 20 to 25%. That much is the influence of genetics. Now, for example, let's say liver fat. We have earlier research that PNPLA3 is one gene which
Okay.
enhances the tendency for fat accumulation in the liver.
Yes.
Uh, of course, brain is another, there is increase in the catecholamine surge through the brain and also various other factors. And of course, the satiety, how much one feels the satiety and so on. So multiple factors, but still, overall, if you see the phenotype, Indian phenotype is worse off, whatever be the factors. And that's why we are developing diabetes and heart disease at an early age.
Thank you, sir. Thank you.
Thank you very much.
Uh,
We'll have to close the session.
Thank you. Thank you, everyone. The session will end now. Thank you so much, sir, for a fantastic deliberation. Thank you.
Thank you.